If you only have 2 plates to do this with, you want to divide your plates into 4 parts. If you have enough that you can only do half a plate, then go ahead and do that. But so I'm going to put 1, 2, 3, and 4 on mine. Obviously, if you have to do all 8 on 2, you want to put another line and mark 4 or 5, 6, 7, and 8, right? If you don't understand, I know you can ask me. It's not very clear. Okay, so I am going to grab some water, and this is where bottled water is best, but if you don't have that, use whatever you've got. And I have some sites written down here, but you can look around. And I've got my fridge door, the door handle. I have an aquarium, and an aquarium always has cool stuff in it if you stick the swab in the water. You can tell I did this in the summer because I had screen door. Needless to say, I'm not using my screen door much, so I'm going to use my other door handle, and I'm going to use my remote, okay, for my TV. You could use cell phones, you could use light switches, you can go check toilet if you want, but pick 8 sites that you want to check. Now, when I do my aquarium site, I don't need to wet my swab because I'm going to put the swab right in the aquarium water. If you were doing toilet water, for example, you wouldn't need to wet your swab. But for any dry site, you want to make sure that you saturate your swab in water. And just like last week, we're going to swab it the same way. So we're going to get our sample. We're going to wet the swab, get our sample, and find the correct area on our plate for that particular number. And we want to swab that area. Okay, so you can go ahead and get started.
Can I swab my dog bowl?
Can you what?
Swab my dog bowl.
Sure, you can swab anywhere you want. Just be careful that you want to know what's in it. It's not always best to know some things. That's actually a good idea. I think I'll do that as one instead of my door. Your swabs, when you're done with them, can go into bleach. Gonna have to find 4 different places for next class. How's everybody doing?
Do you think I would get good results from swabbing my dog's toy? I know I heard a water bowl, but like my dog has an obsession with nylon toys and he has to bring them outside and inside, outside and inside.
Who knows? Try it.
He's a grody boy, so I figured it would be interesting.
Yep. What I find is that a lot of the stuff that you think will grow lots of really disgusting things surprises you. And the places that do grow are places that you would not anticipate. So how's everybody doing? About done? Chiari's done. I can only see 2 people, so I'm going by Shawnetta and Chiari.
I'm all set.
All right.
Almost done.
All right.
I'm done.
Have you ever done like a cleaning sponge?
Ooh, yuck. Yeah, I'll bet that's a good one. I'm gonna do that this afternoon. I have a dish towel or like a dishwashing towel. That's a good one. I'm gonna put that down for this afternoon.
I always wonder how clean stuff gets after I disinfect it.
Dishcloth. I'll try a dishcloth. Who else has got exciting ones that they found to do that I can try this afternoon?
You could do the base or entryway to a cat's litter box.
That's an interesting one too. Yep. That's worth a try. Where?
I'm doing my cutting board.
Ooh, that'd be an interesting one too. I'm not sure I want to do that one though. There, there are certain places I draw the line and I'd rather not know.
I wonder.
Sorry, I didn't catch her. What location was that for?
It was a cutting board.
Ooh, yep, no thank you. I'd rather just not know and live in peace.
Be brave. Yes, exactly. I'm not that brave. Well, there was something I was just thinking of, went fleeting through my mind. But anyway, okay. Well, you guys are probably all about done, if not completely done. And that's all we're doing. So the only other thing that I did want to cover, I know Katie's in the afternoon lab, but if anybody has questions about doing a dichotomous key or about doing your data table, this would be the time to ask. But if you guys are clear on all that and you're moving along fine, then there's no need for us to hang out here. Love you guys, but I've got 3 advisees today in between the 2 labs. So I am perfectly happy to call it quits for today. Have it be a short one.
Hey, Kathy.
Yep.
So for the dichotomous key, that looks similar to what the chart looked like in the micro, the mastering micro, correct?
It would look like a dichotomous key looks like a flow chart. Is that what you're talking about?
Yep.
Okay, yeah, a
Um,
dichotomous key is a flow chart versus the data table, which is where you just put the results for the organism for the test.
Gotcha. Okay. Yep, totally understand. Is it possible that maybe never mind. I can totally just Google it. Never mind. I was just gonna ask for you to send out a visual example just because I'm a visual type of person, but I can totally just Google it as long as
You're gonna say something, Carrie, go ahead.
I was gonna say, I think there's one on the page, I think it's under labs.
It might be under lecture. There is one that I did for gram-negative rods, and I think that one might be under lecture. And it's just a hand-done one that just shows, it was meant to show, students were confused because I had gram-negative cocci, gram-negative rods, facultative anaerobes, aerobes, anaerobes, and so they kind of felt a little overwhelmed with all the different descriptions. So I had done one that just kind of divided things up. That would be an example of it, but I believe your book has a really good example. Also, the notes that I did when we did the Enterobacteriaceae have an excellent one as well. And that I think is from your book.
So is there a set of bacteria that have to be on this somewhere? Because I know you said it was
The
number,
only-
but I cannot.
Only the bacteria that are on the data table need to be on the dichotomous key. So you pick whatever you want to start with. My suggestion is either start with rods and cocci as your first splitting point, or gram-positive or gram-negative. Either of those will probably divide them about in half. And then under that, you can pick whichever tests are on that table, right? You want to use the tests that are on the table because you'll have all those results. And so you then use those tests that are on the table to divide all the rest of them. So eventually at the bottom of this, you'll have 18 individual organisms and you'll have a path down to each one.
Gotcha. Okay.
Okay.
Thank you.
Everybody good with that? Because I know it's for some people, they've used them before, they get it. Other people, it's something new.
It's just setting it up is a little overwhelming at first, but I feel like once I get into it, I'll probably be all right.
And you know what? Once you've got the data table done, the dichotomous key is actually very easy to do. Right, so that's, the key is getting the data table done. And then it's so easy to find ways to separate, because nobody's gonna have the same table. Even if you and Hillary both start separating based on rods and cocci, even if you did that, you'd have completely different table because you'd use completely different tests to separate your organisms. Okay, so there's not really a right or wrong as long as you've got the right results on the data table and you follow them through. Good. All right, well, if there's no other questions.
I'm sorry. I'm looking to see what was on the food and safety lab. When
I'm
we're
glad you said that.
our
Go ahead.
data, we're entering our data and it asks what type of colony, right? Or
Does it?
hold on, let me.
I don't remember that. I don't remember that, but that doesn't mean it's not there. But I'm glad you said that. How did everybody's before and after look with the washed veggie?
Disgusting.
It asks, yeah, it looks gross. It asks what type of microbes. So how do we answer that? Because it
Just
asks.
give just how about a description of the colony?
Okay.
I was thinking it might ask how many, but I didn't realize it would ask.
It does. It has
What
two
type?
parts. On the data sheet, it has two parts. It asks the type of microbes for each one, and then it asks the number of colonies for each one.
Okay, so give a description and
Okay.
then say how many.
Okay.
I'm glad you said that though, because the other thing that when you do all these and you go to examine them, you're supposed to do a hypothesis. So today you want to look at all your different sites and give a hypothesis. What do you think will have the most? What do you think will have the least? And that's part of that lab as well. So you want to do that before you see them all, and because that's the fun part for me, is to see what you hypothesized and how different it is, because it's often a surprise. So you want to do that. The other thing is, if you have anything on these plates this week that looks fuzzy, don't open them. Okay, those fuzzy ones are fungi, and they're probably not going to hurt you. They're around you all the time. But what you don't want to do is have them fly in the air. And if you open them and that's how they spread. So just a cautionary thing. The likelihood is after a couple days, you won't see something like that. But if you wait for a week and read them, you might.
My antibiotic ones had lots of fuzzy stuff on it. And I opened it.
Okay.
Mine did too.
Yeah, I noticed a lot of people's did, which is why I mentioned this specifically now, is because I did notice that people had fuzzy stuff. It's not, you have fuzzy stuff in your refrigerator. You know, when you pull something out of your refrigerator, or you pull the bread out and it's been in there a while and it's got fuzzy stuff, it's not hurting you, but just there's no need to open it, I guess is what I'm saying with these.
Okay, so don't pour the bleach in it, just.
Oh, you'll pour the bleach in it, but I mean, when you're looking at it or taking pictures of it, just do it through the top.
Oh.
You don't have to do this and, examine it for description or anything like that. You can say fuzzy and leave it.
Kathy, I decided to do an interesting one, or one that I thought would be interesting. So I didn't have any lettuce or anything in my house. And the next day was grocery day, but I was like, what do I have? And I had a potato. So I did an unwashed potato versus a washed potato. It is something.
Yeah, it always reminds me how important it is to wash things when I do that particular one. So
Yes,
my
I will
milk-
be using a
my
scrubber
milk-
brush now.
Oh, yeah. My milk wasn't that impressive. I don't know about you guys, but
No, there wasn't much difference in mine either.
Yeah, in mine. I have a question though. This was on the lab materials for today, the
Yep.
sodium oxide. So
yep, remember, that's the one that I mentioned as, you could do it. It's the second
Right.
half of this lab or not. And it is fun. If you have more than like 2 or 3 people, I mean, not little kids, I wouldn't do it with little kids. But if you have a couple other adults or teenagers in the family, it is an interesting one because it really gives you an idea of the spread of something, but I'm not going to expect you to do it. So that's up to you.
Okay. Perfect. That's all my questions.
All right. Well, if you guys are good, I'm good. So if there's no other questions, you guys have a great day.
Have a
Thank
great day.
you. Bye.